My talk comes with a bit of a preamble. First, I just want to say I'm really excited to be here at NIMBioS, which apparently I'm saying wrong. NIMBioS. Thank you. It's really nice to be here, and so thanks to NIMBioS for funding this visit. It's very exciting. The second preamble that I have is, it's been a while since I've given a seminar-style talk. I just had my first teaching assignment start in January, so if I'm particularly spazzy, it's because I'm used to talking to, undergrads. And they seem to like when I'm spazzy. Right, so Judy stole some of my words from this first slide, which is just to tell you basically that my research combines tools from mathematical and evolutionary biology to understand something about infectious diseases. And really, there's sort of one big question that kind of most of my research can fit underneath, and that question is, why do some parasites cause severe disease while others don't? If we look at the kinds of disease caused by different parasites around there's tons of variation. So even within a single kind of parasite group, there's tons of variation in the severity of disease caused by these different parasites. And that's true if you look across species.
It's not on.
It's not on. No. It is on. Green light.
Oh.
No, your hair. My hair. Oh. Right, sorry. I didn't realize this was a modeling talk. All right, so, okay, good? We're good. Awesome. How do I look, guys?
Excellent.
All right, so totally derailed. So there's tons of variation if you look across species. There's variation if you look between closely related species, and there's variation even if you look within a single species, and that's what I'll talk about today. And we know a lot from biomedical research and our friends in immunology and parasitology about what leads to disease. But that really is, that kind of research focuses on disease per se, rather than maybe the interactions that are giving rise to that disease. And so I think often what is overlooked is the fact that parasites are responsive and complex critters, just like other organisms, and they're often not treated like this. And I think evolutionary and ecological research can add this to disease research. And so my research, I'm trying to show that we can apply tools from evolution and ecology to gain new insight on disease. And I see this question kind of breaking down into three different sub-questions. And the first one is the proximate questions here. So what is it in particular about the ecological interactions between hosts and parasites that give rise to disease? We've got the ultimate sort of questions here. So why are parasite traits the way that they are? What has caused them to be this way that leads to a certain severity of disease? And finally, given that there is all this variation, and variation is the raw material of evolution, can we predict how parasites will evolve and diseases will change in the future? So if I have time, I'm going to try to speak to all three of these parts. Right. So first part, understanding the ecological interactions. What is it in particular about the interactions between hosts and parasites that give rise to disease, and what leads to variation? Every story I'm going to tell you today is going to be about malaria. So here is the requisite malaria life cycle. Key points for this talk are that the malaria parasites spend some of their time in a mosquito vector and some of their time in a vertebrate host. And from the perspective of understanding disease severity, this is the bit of the life cycle that we care about. So, I'm terrible with pointers because my hands are really shaky, and it's not on. Problem number 2. There we go. Okay, so parasites enter red blood cells, they replicate inside those red blood cells, eventually bursting the red blood cell, releasing those progeny parasites, which can then go and infect another red blood cell, continuing this phase of red blood cell invasion. That's the key point. This is what causes disease. Hosts get really anemic. You're bursting red blood cells. This is a problem. Another key point is that sometimes instead of following this cycle, an infected red blood cell will go on to produce a transmissible stage of the parasite called the gametocyte, and that's going to be relevant a little bit later on. But for right now, what causes disease is this cycle of invasion, replication, and bursting, and that's what I'm focusing on. So I sort of hinted at the fact that there's variation in disease outcomes across different strains of malaria parasites. This is true at the species level as well. So there's 5 species of malaria parasites that can infect humans, and we see tons of variation in the severity of disease. So Plasmodium falciparum is the most well-known malaria parasite species, and it is the cause of the most mortality and morbidity from malaria. But if we look even at a smaller scale, so not species level, not across species, but within a species, we see variation as well. So this is some data from experimental rodent malaria infections where we control as much of the variation as possible. These are inbred lab mice, fed the same diet, yada, yada, yada. Tons of variation controlled. The only thing we try to vary is the genotype of parasite that's infecting the mice. And so here we have two measures of the outcome of infections. We've got how anemic the host gets, how many red blood cells they lose. So over here, really anemic, pretty severe infections. Over here, not so anemic, not so severe infections. And then another measure is the cumulative parasite density over the first wave of parasites. So these points here represent, each point is a parasite genotype, each color is a different parasite genotype, and each point represents 5 replicate infections in mice. So what you see here is there's variation in severity, and that genotypes tend to cluster close together. The same genotype clusters close together on this graph. It's just the absolute reduction from where it started to the baseline, as low as it gets. So that's some measure of outcome, and then on the right here we've got some measure of the dynamics of these parasite densities over time. And again, each color represents a different genotype, and again we've got 5 replicate infections, means, and standard errors. And what you see is that different parasite genotypes give rise to different dynamics, and that those dynamics are pretty repeatable across individual infections. So we know that there's this variation, but we don't really have a good idea of why some strains are more virulent than others, and why the patterns are different. But we've got tons of this sort of dynamical data. And so this is an interesting starting point to ask the question, can we use these dynamics to infer the interactions? And I have this slide here. This was for the EEB folks, and I heard that maybe there weren't so many of them that were going to make it here today. Just to make the point that we've got this huge body of tools that uses dynamics to infer interactions. That's theoretical ecology more broadly. Then I like to do this sleight of hand where I go, look, it's just a predator-prey model, but with different players. So we can use these same tools to understand within-host infection dynamics, and for malaria, the key resources are these red blood cells. We might want to think about immunity being the guys that are killing the parasites. And so what I want to know is I want to use a sort of modeling framework to ask what are these important interactions, how important are these different elements, and where and how do these interactions differ across parasite genotypes. And in particular, is the variation due to parasite traits that have something to do with exploitation or replication? Is the variation to do with host traits, like how they're producing red blood cells or how the immune response is interacting with these different strains? Who's controlling the severity of infections? And the nice thing about experimental systems is that they can be manipulated. So when I was a PhD student, this much complexity confused me and scared me. So I was afraid of immunology. Sorry, Nathan. As a PhD student, it's growing on me, I guess. But we had some data from a kind of simpler system. These were immune-depleted mice. So what we wanted to know is, can we ignore immunity completely, understand the dynamics of what's happening in the system, understand the differences in dynamics across parasite strains, without having to think about immunity at all, just thinking about resources. And we have these 2 parasite genotypes. Green one here, which throughout the talk is the more virulent one, and the brown one, which is the less virulent one. And I should say that I'm using virulence here really as a shorthand to mean more and less severe infections. But this is kind of giving you the idea that maybe I'm treating it as an intrinsic property of the parasite, because I use this word really sloppily. So one important question is it an intrinsic trait of the parasite? Again, is virulence related to these exploitation traits, or is virulence a product of the parasite traits and the host responses? Right, so I want to build a model to understand these dynamics, and I like to start as simple as possible. So this is the simplest possible description of what's happening in a malaria infection in a mouse. So you inject some parasites into a mouse. These blue guys here are our parasites. The red guys are our red blood cells. Really quickly, those parasites have to infect a red blood cell, or else they'll die in the bloodstream. We can imagine that we've got some red blood cell turnover. Just this is what happens naturally. The host has some homeostatic equilibrium that is trying to maintain of red blood cell densities. So old ones die, new ones come in, and I'm treating these new ones separately, so they're bigger and darker, and that'll be relevant later. Then the next step is we have the parasites replicating in those red blood cells, and eventually bursting the red blood cells and releasing the progeny parasites, which can start the cycle again. And what's really nice with rodent malaria is that this whole process takes 24 hours, and it's synchronous in an infection. So all the parasites go through this cycle at the same time. They all burst at the same time. So I can model this in a nice discrete time framework. And this is the simplest, I think, possible description of that biology I just showed you. So we've got parasite densities over time, red blood cell densities over time, and all you need to know is to know how many parasites there are going to be. You need to know how many red blood cells got infected the day before and didn't die, and then how many burst out. How many burst out from an infected cell? For red blood cell dynamics, we need to know how many red blood cells remained uninfected, and then we need some production of new red blood cells. So this is the simplest possible model, and I can add complexity in here in various ways. And I'll show you a few of the ways, I'll show you all of the ways that I added complexity into this really simple model. Right, so I'm completely ignoring immunity, and I'm just asking, can resource availability and maybe resource specificity define what's going on in the dynamics? So one way to alter how resource availability works is to think about how the host is producing new red blood cells. So we imagine that as you get more and more anemic, you're trying to, as a host, you're trying to produce more and more red blood cells to make up for that deficit. So that's what this shows. As you get less and less red blood cells in your system, you produce more in a given day. It's a nice linear function, easy to model, but maybe there's some sort of cap on how many you can produce. Maybe, more importantly in my world, is maybe when you get really anemic, you can go into sort of overdrive and produce extra red blood cells to make up for the extreme anemia you're experiencing. So rather than using this saturating function, what I actually used as a PhD student, because 3 parameters was too confusing, I used sort of a piecewise linear function where this was a steeper slope than this part over here. Another thing that can potentially alter availability is the production of these transmission stages. So some fraction of all infected red blood cells are going to go down this route, making the transmission stages, which do nothing in a host. They just circulate, hoping to get taken up by a mosquito. And then these guys, the rest of the infected red blood cells, go through this cycle of replication and bursting. And these are the guys, of course, that can then go and infect new red blood cells. So all else being equal, the more you shunt into this pathway, the fewer red blood cells you're going to infect overall. Then I wanted to think about maybe resource specificity is important here. Maybe parasites treat different ages of red blood cells differently. And there was some evidence from earlier models that maybe this is important in infection dynamics for malaria. I just covered up that mic for a second. Sorry, guys. Right, so reticulocytes, when young red blood cells enter the bloodstream, they're actually physically quite different. They're bigger, they still have some intracellular stuff in them, and the surface proteins, surface receptors, I think, are actually quite different. And it takes about 3 days for them to mature and expel all their intracellular stuff. So I think actually a parasite might see them as being quite different sort of resources. And there's 2 ways I thought about them being different. One is that parasites might have a harder time invading one of those types of cells than the other. And the other option is that they're fundamentally different environments. And so how much replication can go on in those different cell types might vary. So adding all this complexity leads to a slightly more complex model. And what I like to say on slides like this is, I didn't put this here for you to understand it. I put it here to impress you, which I feel is especially important in this room. Right, so now we've got this age-structured model where we explicitly track different ages of red blood cells. We've got our young ones, which last 3 days in the bloodstream, and then our fully mature ones. And you can get back that original simple model just by setting some of these parameters to 0 or setting them equal to each other. So really I looked at every combination of those different things that I told you about and asked which of those different biological hypotheses best explains the dynamics that we see. Right, so I fitted each of those models to some data. We had 13 different data sets, so different individual mice infected with parasites. So in my lynx and hare analogy, that's like having 13 different forests, which is nice. So I fit the models to every individual rather than means. What this is showing you here, data are in the points and the dashed lines. The best model fit for a given model here is given in the solid lines. This is one mouse. This is the simplest model, and I fit only to the red blood cell dynamics. So I kind of used the parasite output as kind of validation of the model in some sense. This is the simplest model. Here's some increasingly complex models. And then I statistically compared the fit of the models to the data. And for this individual, this is the best fit model, and you'll see that the parasite outputs are actually reasonably good. They're capturing well the dynamics of the parasites, even though I didn't fit them to the parasite dynamics at all. And in fact, you go through this for every mouse, and we ended up with a single model, which was the best predictor of dynamics in every case. And what we learned in general was that age structure was important for explaining dynamics, and how red blood cells were produced was important. Having this variable production with anemia was really important to explain dynamics in general. But in terms of what explains the variation in dynamics with different parasite genotypes, there's a few things we found. First, the more virulent parasite had higher burst sizes, so the number of progeny parasites produced was higher, and this was especially true in those young red blood cells. This guy seemed to have some sort of advantage of infecting young red blood cells, where this one did worse in young red blood cells. Second, we found that the more virulent parasite had faster invasion rates of mature red blood cells, and both genotypes seem to have a hard time infecting the young red blood cells. Oh, and that was it. Sorry, I forgot that I was it. What's really interesting about these sort of inferences, so I worked with Andrew Reed, and he was on this paper with me, and he said, these are the sorts of things that you can't just look at the data and see. You can't look at this data and go, oh yes, obviously this has higher invasion rates in mature red blood cells. And so this is why I think modeling is really key for gaining new insight that is just not available from looking at the data. So this sort of variation in parasite traits between the more virulent and less virulent genotype could explain dynamics that we saw in competition. So you put these two strains in a single host, get some dynamics. What I did here to get the model outputs was just take sort of the median, the best fit parameters, median best fit parameters for each genotype, plug those parameters into a model, and then spit out some dynamics. So there's no fitting going on here, it's just prediction based on means or medians. And so I think this looks pretty good. So we've got our red blood cell dynamics here. This is one individual data set. We've got 5 data sets that all look pretty good. Red blood cells over here, we've got the parasite dynamics, and the model does a pretty good job of capturing what's happening. It kind of falls apart in the second peak. We're predicting too much competitive suppression from the virulent genotype. So we took this, the fact that the model could sort of capture or predict something in an experiment where we didn't fit the model to that data, gave us some confidence that we were capturing the right elements in the model. But the way that I feel, and presumably the way that people in this audience feel, is that math doesn't define biology. It kind of offers plausible explanations that ought to be tested. And this doesn't sound surprising probably to you guys. This sounds like the scientific method, but I feel like in disease dynamics this actually happens pretty rarely. So I really wanted to test these predictions. Is it true that the more virulent parasite has higher burst sizes? Does it really have faster invasion rates? So I went to some trouble to develop new empirical approaches to measure these things in our experimental system. And I like putting this picture here too, because I didn't spend that much time counting little purple dots under a microscope, but I have like incredible sympathy for people who do this as their only job. It's not fun. Oh, and the beauty of math is that it's so certain. Counting purple dots is really scary and error-prone, presumably. Anyway, all of that aside, here's what I learned. I found that indeed, the more virulent parasite did have higher burst sizes. This is just mean burst sizes estimated, complicated experimentally, but we found support for this idea. What was interesting is we couldn't find support for this idea that they had especially high burst sizes in young red blood cells. That didn't seem to hold up when we did our second experiment to test these predictions. We also couldn't find any evidence that invasion rates were different across parasite genotypes. And also we expected these invasion rates to be way down here. We expected them to be about an order of magnitude lower than we measured them to be. So no evidence of that either. So I could have just written up this experiment as a separate paper, but what we're left with is, okay, some of our model predictions are hold up when we experimentally test them, some of them don't. So what does that mean? Does that mean the model's too simple? Should we have thought about including immunity in here? Does it mean you shouldn't let a theoretician do an experiment because they're going to screw it up? I'm not sure. So we have a whole new data set because we basically repeated the whole experiment, so we've got a whole new set of dynamics for more individual infections. New data, new models. I worked with Nick Saville, who's a card-carrying Bayesian, so he helped us devise some new fancy approaches for testing differences between strains using hyperparameters, which I think are quite cool and exciting. And we added immunity into our model, and we added immunity in what I think is a sort of simple first approach way. I think often models of disease dynamics make really complicated assumptions about the mechanisms of immunity. What we did here is basically we did it phenomenologically. So we have a killing rate. Immune responses turn on, they start killing parasites at some rate, and then immune responses turn off. What I'm showing you here is the best model fits for every individual data set again, and the key thing to take away from these pictures, details aren't important, but the model fits are clearly way better than they were from the original model. So, what are our new inferences? Well, the new model tells us that burst sizes are different between the virulent and the less virulent genotype. So, now this prediction has survived 2 theoretical tests and 1 empirical test. So I think we have pretty robust evidence that these burst sizes are different. The new set of theory tells us there's no difference in invasion rates. This matches with what we saw experimentally, so perhaps there's no difference in invasion rates. And finally, what was quite interesting is that we needed to have immune responses. We needed differential immune responses to recapitulate the dynamics. What's really interesting is the infections with the more virulent genotype, you needed immune responses to recover the dynamics. With the less virulent genotype, you don't need immune responses to get the dynamics in that system. So that suggests something quite interesting to me. One, virulence, whatever virulence is partly controlled by parasites, has to do with burst sizes and their exploitation traits, and it's partly to do with host strategies for defending against parasites. Right, so some general thoughts. What have we learned here about the biology? I think resource limitation is a major driver of dynamics in this system, but immune responses, even in these immune-depleted mice, seem to be playing a role. And like I just said, in terms of who controls the severity of infection, it's partly parasites, it's partly hosts. Which maybe you could have told me before, but here we've got something slightly more specific about which traits it is of the parasites. Generally, I think this shows that this iteration of theory and experiments is quite important for generating actual understanding of a system. And also, I haven't made this point yet, but intuition biology is not particularly rigorous. So one thing that biologists think is that these secondary peaks of parasites are due to antigenic variation. So it's due to parasites changing their coat proteins and evading immune responses. My models show that you can get these secondary peaks just from resource dynamics alone. And the thing is, parasites are constantly changing their coat proteins, so that secondary peak would be antigenically distinct even if it had nothing to do, even if that wasn't the reason why there was a secondary peak. And I think I've already said this point, but math can reveal biology that's not obvious from data, and math is awesome. Right, so that's a bit of ecology. Moving more into evolutionary stuff, I'm really interested in understanding why parasites do what they do. What explains the evolution of their traits and behaviors? And life history theory is a powerful theoretical framework for understanding traits of multicellular organisms, and I'll show you that it can apply to parasites as well. So here's our malaria parasite cycle of replication in host red blood cells. Again, I told you that sometimes these infected red blood cells go on to produce these sexually differentiated gametocytes, which are the form of the parasite that, when taken up by a mosquito, generate an infection in a mosquito and ultimately an infection of a new host. So parasites have to decide how to allocate their red blood cell resources between the sort of cycle of asexual growth and reproduction. And this has a clear analog in multicellular organisms between growth and reproduction. So this is like the growth of an infection, the maintenance of an infection, versus reproduction or generation of new infections. So it's obvious that organisms need to balance these two things, balance their investment in these two things. But what's really strange is that it seems that malaria parasites actually invest very little in this reproduction or transmission side of the balance. In fact, something like less than 1% of all infected cells go on to produce these gametocytes. And it's especially surprising when you consider that these guys are producing vast numbers of these host-damaging asexual cells, asexual parasites. So all else being equal, the more you invest, the less you invest in gametocytes, the more you're investing in these damaging parts So thinking about how parasites allocate their resources between these two paths might tell us something about virulence evolution as well. So in evolutionary epidemiology, we've got this metric for parasite fitness, for disease fitness, and that's R0, the number of new infections produced from a single infected individual in a wholly susceptible population. And this is basically often thought to be the standard measure of fitness for diseases. And an expression for R0, you can derive it from a model of between host dynamics. So for malaria, what you need to think about is all of the processes that go getting an infected person to infect a mosquito, and then for that mosquito to infect another person. And the details of this R0 expression are not terribly important. On the top here, we've got the gain of new infections via mosquito, so you have to infect the mosquito first and then a new person. And down here, we've got the loss of infections, whether they be human infections or mosquito infections. So we know that evolution maximizes this quantity. So to understand how parasites allocate their resources between growth and reproduction, we have to understand how allocation affects R0. So you can use an evolutionary invasion analysis to ask how do allocation patterns affect R0. And it's going to affect it through various parameters of this model. So the first thing could be mosquito mortality. If you produce too many gametocytes, and those gametocytes, too many gametocytes get into a mosquito, there could be some effect on mosquito longevity. And the data on this is kind of conflicting. It's not clear whether this happens or not, but it's something that's been suggested out there in the literature. So mosquito mortality pops up in here twice. One, because I said this was loss of infections, so loss of infections because mosquitoes die. And then up here in the gain of infections because mosquitoes have to live a certain amount of time before they become infectious to a new person, and that's about 2 weeks. Before I tell you whether this can drive the evolution for low investment in gametocytes, I'll tell you another thing that's been suggested in the literature. One is transmission blocking immunity. So if you produce too many gametocytes, you might stimulate an immune response that targets those gametocytes. So maybe having too many is bad from a perspective of actually getting into a mosquito in the first place. Right, but what we find in both of these cases is that there might be selection for having low numbers of gametocytes, but that's not the same thing as having selection for low investment in gametocytes relative to asexual production. And I'm going to try to explain this with graphs that I just made last night. They're really simple. I hope they make sense. So if you think about you're a parasite, down here you've got what proportion of infected red blood cells you're going to allocate to gametocytes, and then how many cells you infect over the course of an infection. So if you allocated everything to gametocytes, as soon as you get into a host, you infect some red blood cells, and you convert them all to gametocytes, your infection is over. So you're down here somewhere. You infect very few cells. If you allocate nothing to gametocytes, you end up infecting a lot of cells. But numbers of gametocytes are the product of how many cells you've infected and this rate of conversion to gametocytes. So you get this sort of hump-shaped curve. So both of these hypotheses tell you that there's some number of gametocytes which might be optimal, so I'll just make up some number. That's your optimal production. But there's two ways to get that production. One is infect very few cells and convert them all to gametocytes, or convert a high proportion of them to gametocytes, or infect a lot of cells and convert very few of them to gametocytes. So the idea is you can sort of change your growth rate to change the slope of this blue line, which just gives you a slightly different hump-shaped curve, but there's always at least two ways to get the same number of gametocytes by just changing how many red blood cells you're infecting versus how many you're allocating to gametocytes. Does that make some sense? Okay. It's not, yeah, good. I just made that up. So, right, it could be. The thinking here was, so it's possible that that could be the optimum, but I think the thinking here is that these two hypotheses predict some low level of gametocytes. Too many will kill a mosquito. So unless, yeah. Right. Right, so it could be the case that this many kills a mosquito, in which case that would be best. but making an assumption that, somewhere up here it still kills a mosquito, so you're best to keep that proportion low. Obviously the math is way more explicit in the actual paper. Right. Okay, so those were some suggestions that were in the literature, but these decisions about allocation are happening within a host. And so it's important to think about what makes parasites successful within a host and what sort of challenges do they face within a host. And one of the challenges they face is multiple infections. So for malaria, having an infection with multiple parasite strains is quite common. Where malaria is endemic, it's something like 70 to 90% of infections harbor multiple strains. So these strains are clearly going to be doing some competition within a host. And thinking about these within host processes changes the R0 expression into something that looks a little bit messier. Within host competition, it comes out in these parameters here, and you can show analytically that what within host competition does is generate selection for low investment in gametocytes. So all else being equal, in competition, parasites that secure more resources do best. So if you can have more of the red blood cells to be your own, you're the parasite that's going to win the within host competition. And so this generates selection not for low numbers of gametocytes, but low allocation to gametocytes, which is what we were looking for. And Laura Pollitt was a PhD student in Edinburgh who sort of tested this hypothesis that competition can generate selection for low investment in gametocytes by taking different parasite strains, putting them in an infection by themselves, looking at how they allocate the resources between asexuals and gametocytes, and then putting them in mixed infections and seeing what happens. So the solid lines here, again, different colors are different parasite genotypes. Solid line here is that genotype in an infection by itself and what proportion they're allocating to gametocytes. Dashed line is what happens when you put it in with competitors. So in every case, parasites are reducing their allocation towards gametocytes. They're responding to the presence of competitors in the way that you'd expect from the theory, reducing their investment in gametocytes so they can make more of those asexual stages which are gathering up the red blood cell resources. So the within host environment definitely alters these patterns of allocation. And there's some sort of interesting side notes to this. Parasites adjust their allocation in response to drugs as well. And there seems to be some variation. So really strong drug treatment, which is probably going to clear an infection, parasites seem to sort of do this terminal investment strategy where they shunt everything towards gametocytes because they need to get the heck out of that infection. Sort of low levels of drugs, subcurative doses of drugs, seems to shift parasites into this reproductive restraint mode where they say, okay, let's not make any gametocytes, let's hunker down, maintain enough asexuals that we can keep this infection going. And we're really interested in looking at predicting the optimal patterns of allocation to gametocytes in different environments and different levels of stresses. Can we figure out what that picture should look like exactly? When you vary the stress, how should allocation vary? And there's been some work done on this by some other folks in the past. We're just trying to tie it more closely to some data that we have. Right, so the last thing that I want to talk about, I think I have some time, is evolutionary responses. So this, I feel like this is like the money stuff. Sometimes when I talk about evolutionary responses, I talk about my work on drug resistance or developing new theory for studying disease evolution. But today I wanted to plug some work on circadian rhythms, sort of. So the link to this evolutionary responses is a bit weak, but bear with me. I wanted to talk about this stuff because it kind of relates to stuff that I'm here to work on with Judy and Ed, and it's kind of cool. Right, so I mentioned this idea that all the parasites in an infection are synchronous. They're all bursting in unison, they're all infecting red blood cells at the same time. And we've known this for quite a long time. Here's, again, the different species of malaria that infect humans. This cycle, I said for rodent malaria it took 24 hours. For human malaria, it's always a multiple of 24 hours. Falciparum takes 2 days to burst. So this gives a suggestion that there's some sort of circadian basis to this life cycle. We've known this for a long time, but we haven't really known why. So people used to use the periodicity of fever as a diagnostic tool. So you get a whole bunch of parasites bursting out at once. This generates a fever response in a host. If that fever is every 2 days, you know it's one of these species of malaria. If it's every 3 days, you know it's a different species of malaria. So we've known this for a long time, but we haven't really had a good view on why parasites would do this. And you can think of lots of reasons why you wouldn't want to do this. All else being equal, you get all these parasites bursting out at the same time. You're maximizing competition for red blood cells, presumably. An interesting further point is it's not just that these parasites do this all at the same time, it's that they seem to do it all at a particular time. So for the rodent malaria species, they're all bursting out at midnight. So whether it's the timing that's important, whether it's the synchronicity that's important, whether it's both of these bits that are important, is not clear. Whether it's important at all is not clear. But I'm going to tell you what my best guess is, why parasites might want to have this timed and synchronous production of parasites bursting out of red blood cells. This really complicated picture shows you some of the main components of the immune response that fight malaria. And what has been shown is that many elements of this immune response seem to be regulated, have this circadian regulation. So these are gene expression studies showing that gene expression follows the circadian rhythm, and this is different elements of the immune response again. All of this work, oddly so far, has been done in the absence of infection, or at least malaria infection. So we know that immune responses have this rhythm when they don't have malaria parasites to face, but we're faced some sort of general challenge. We don't know whether these rhythms hold up in a malaria infection. But my thinking is, if immune responses have this circadian expression, parasites might want to alter their developmental schedules so that they keep the most sensitive stages of development away from the peaks in the immune responses. This is all like super speculative right now, but we're working on the problem. So me and a few other folks have an HFSP grant to study circadian rhythms in host-parasite interactions in malaria infections, and we're really looking to tease apart, well, when are the key immune components against malaria, when are they active, what are the most susceptible stages of the parasite, do those things match up, or are they sort of like, are their schedules off? We really want to be able to define the costs and benefits to the hosts of having these rhythms and the costs and benefits of parasites. We want to know whether the hosts are really driving this rhythm or the parasites are driving this coordination. And importantly, we want to know what's limiting parasite developmental schedules. So can they adjust their schedules so they're bursting at any time of day, or do they have to, for some reason, burst at midnight for rodent malaria? And the reason why I think this is relevant and interesting is because resistance to frontline antimalarial drugs is thought to be mediated by altering their developmental schedules. Parasites that are able to delay the replication might do better in drug-treated infections. And so I think it's really important to know what are the limits of parasites altering their schedules. Can they actually, are they free to alter their schedules at will, or are there some limits on when they can, what are the limits and constraints on when they can do the replication and bursting? And if we can understand what these costs and limits are, we might be able to predict the limits to parasites changing their strategies in drug treatment. It might give us new insight into better ways to administer drug treatments or new insights into drug targets. Right, that was my tenuous link to consequences, but I think it's some exciting stuff. So just a quick summary of what I've told you. I've showed you, hopefully, how we can apply population dynamic models to host-parasite interactions to identify the host and parasite traits that govern disease. I talked about applying life history theory to parasites and showed you that parasites have the same sort of life history trade-offs as multicellular organisms, and this might be relevant. I mentioned briefly that parasites seem to adjust their allocation in drugged environments, so that's going to have some implications for health, obviously. Finally, understanding the costs and limits to parasite traits is important for predicting parasite evolution. Quickly say thank you to some folks who helped along the way and some funding. If you have any questions, I'd be excited to answer them.